Okay, good morning, everyone. Welcome to the last class. We've made it. We've almost made it. We have one more class next Tuesday, bright and early, 7:30 in the morning. Yesterday. On Tuesday, I went through all the expectations for the final project, written report, oral presentations, stand up on the right side of the screen, move to the left side of the screen. We don't need to go over any of that again, I don't think. Please review all that material in the recorded lecture from Tuesday's recorded lecture if you're not clear. Just again, a final reminder, we've got 60 presentations. They are all awesome. We're going to try and we will get through all of them in 2 hours and 45 minutes, as long as everybody keeps to length limits, submits publicly viewable YouTube videos at the right place at the right time, and so on. So please do your part to make sure we run a well-oiled machine Tuesday morning. Any questions or anything that wasn't clear from last time?
In the report, if our plots are too big, for example, like we put them in an appendix.
You can put them in an appendix. That's fine. If you've got a lot of visual results to report, that's fine. Let's keep the prose to about 4 pages, please and thank you. Yes.
The video can just have no audio.
The video should have no audio. I will mute things anyway, so if you want to add a soundtrack, knock yourself out, but we will not hear it. Yes. Yes.
So, you said for the second part of the final report, you want to describe our code. Do you want pseudocode? Like, how would you want us to describe it?
You're describing the functionality, how you implemented things. Where did you make changes? You can include pseudocode if you want. We just want you to try it. Assume we don't remember exactly how you did things. It's probably a pretty good assumption because there's 60 of you. So high level view, what were you trying to do? So we're about to read the results, right? You're A and B, and we are going to try and assess whether it was a fair comparison. Is the hexapod 10 times bigger than the quadruped? If so, probably the hexapod is going to win in your A/B test. So give us enough description to understand how you attack the problem, how you implement it. Describe for us enough detail to see that it is a fair comparison between A and B, that level of detail. If you want to include pseudocode, that's fine. Use common sense. What we don't want to see is, here's a screenshot of our code. Doesn't mean anything to us. Yep.
For the presentation, you just want us to walk through a brief run through your paper?
The oral presentation?
Yeah.
No, the oral presentation is not a scroll through of your PDF. It is the opposite, right? You want to, in your 2 minutes and 30 seconds, show us what you did. Don't tell us what you did. So video, fitness plots, footprint graphs, phylogenetic trees, not as much, but focus on the visuals, right? This is just true of verbal communication and science communication in general, right? You could create a slide deck with a whole bunch of bullet points and read them off, right? I have tried to not do that in this class. You've sort of seen how I've tried to communicate to you a lot of information visually. Verbally, the oral contribution is a complement to what's on the screen.
When showing how the robot can evolve, you're recommending like a blind comparison that we could do between like the different groups and like maybe like a control. If the control was not like, if it was like just evolved for like less, like a smaller amount of time on like not as like specific of like the conditions of your robot or like a fitness function, would that be like a fair comparison, or does it like sort of have to be trained in like the same number of generations to actually like have a fair comparison?
That's a good question. So it's a question about what makes for a good control. So you were trying to achieve something in your final project. Look, I achieved it. How do we know that you achieved it? Usually it's the robot is jumping better, it's walking better, it's hitting the ball better. Better than what, right? The what is the control. What counts as a good control? For the purposes of this class, a control is a random controller. Show us 2 videos simultaneously, one with your robot being controlled by a random controller, so a random set of synaptic weights, and the other video same robot, same task, same physics engine, but it's being controlled by the most fit controller or the most fit robot body that your evolutionary algorithm found. And if we can tell, without you telling us, that's what makes it a blind test, if we can tell which is the evolved and which is the random, that's pretty good evidence that your evolutionary algorithm actually did something. It was able to improve fitness significantly above what you would find in the initial random population.
Isn't the goal also to have that difference between the A and the B?
Okay, right. So basically there are two pairs of videos we really want to see in your presentation, right? The first pair is what I just described. Show us that your evolutionary algorithm worked. Whatever it was, whatever that evolutionary algorithm was. Did it improve significantly over random behavior and random robots? The second pair of videos is, here's some robots from A, here's some robots from B. And if the result or the claim you're trying to defend is A is better than B, then the same thing. We should be able to immediately see that A is jumping better than B or A is running better than B, whatever it is, if that's your claim. If your claim is B did better than A, then obviously we should see the behaviors of B are better than the behaviors of A. It is also fine in your oral presentation and your written report to defend the claim that there is no difference between A and B. The evolutionary algorithm improved behavior in A, and here's proof that evolution made progress. Here's proof that evolution improved things in B as well, but there's no difference between A and B. That's also fine. It's the fourth outcome you're trying to avoid, which is here's some behaviors of A and here's some behaviors of B, and it's really hard to tell whether one is jumping better than the other. I don't really know whether what I put into my variant B of my code actually made it easier for the evolutionary algorithm to evolve, jumping, walking, hitting, whatever it is. Make sense?
So, for the report and the pictures count for a page count, or is it just for words?
I think you can put it in an appendix if you want. If you've got a couple of figures, then that's fine if it's in the body of the 4-page PDF. But if you've got 4, 6, 8, 10 figures that you want to reference in your document, put them in an appendix and reference them in the main body of your report.
If our computer can exactly handle like many generations of runs, and then for a fitness graph, it wouldn't exactly be like a nice curve or anything. It would kind of be like a small juts. That'd be okay.
That's fine. So again, we're assuming that not everyone has the same high-powered laptop. You want to be able to do enough evolution so that there's a difference between random behaviors and evolved behaviors. So it doesn't need to be a smooth curve. The way you would get a smooth curve is if you did 10 independent trials of your evolutionary algorithm. You rewind the tape, run it again with a different random population, you get a second fitness curve, rewind the tape, third curve, rewind, fourth, rewind, fifth. And if you average all of them, the more of them you have, the smoother the curve you're going to get. But if your laptop is entering its golden years, you might not be able to get as many, so you might have a more rugged looking or discontinuous fitness curve. That's okay, as long as the end of the curve, wherever it is, 10 generations, 100 generations, 1,000 generations, as long as it's significantly higher than where you started in the initial random population in generation 0. There are no recommendations in this class about how big your population should be, how many generations you need to run it for. It's up to you. It depends on what you're evolving and what you're evolving it on. Other questions? Okay, there were a couple of good questions about the format of the oral presentation, how to do this well. Again, I'm just going to repeat what I just said. I think most of you have probably never given this short an oral presentation before. 2 minutes and 30 seconds is a surprisingly difficult thing to do. As the instructions for the final project say, there's a lot you need to communicate to us all in 2 minutes and 30 seconds. My advice is write out verbatim exactly what you plan to say. it'll probably be, if you read what you just wrote in your draft, it's going to be more than 2 minutes and 30 seconds, I'm going to guess. Go through your text. Everywhere you can replace 2 words with 1, do so. Then go back, do it again, do it again. Tighten your oral delivery of what you're going to say until you get it under 2 minutes and 30 seconds. And then practice, 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 practice. Someone was asking about whether there's going to be a timer in this class. There is not. We're going to run a YouTube video, so the only thing we'll see is your video. And I apologize, there's no clock in this room. I'd bring one, but in my personal experience, trying to watch a clock and mentally estimate where you are in your presentation and speed up and slow down, you don't have time to do any of that conscious processing in 2 minutes and 30 seconds. The only thing you have time to do is memorize what you're going to say and practice it so that it's 2 minutes and 30 seconds, so you don't have to consciously think about timing and presentation while you're speaking. You will not have time. Okay. That being said, as you make your oral presentation, if you want to put a few visual cues in there to remind you of exactly where you are and what you need to say, that's fine too. But again, please try and resist putting a whole bunch of bullet points and then reading off those bullet points for us as you go. Yeah. Okay, another good suggestion was how to make this 2 minutes and 30-second presentation. I suggested OBS. It's a good way to record a bunch of videos. You can get the timing down to 2 minutes and 30 seconds. You can make a slide deck and embed videos in the slides and then advance those slides as you're speaking and screen record that with no audio. And then you know that the advancing of the slides matches what you want to say verbally. Yeah, just a couple of strategies, suggestions. Anything else about how to prepare this oral presentation? No? Okay. All right, so we are finally at the final lecture for this course. We are continuing on our theme of evolving bodies and brains. We started in the mid-90s looking at Carl Sims' Evolving Bodies and Brains of Rigid Robots. We moved forward into the last 4 or 5 years, where Professor Cheney and some others have started to evolve bodies and brains for soft robots. We looked at the how to evolve bodies and brains of soft robots. We looked at the why of soft robots, why you might want to evolve and deploy autonomous machines that are made up of soft and rigid components. Today, we're going to look at evolving biological robots, which have now become known in the popular press as xenobots. Some of you have probably heard of xenobots before, so I've sort of given away the punchline already. But going back to sort of the overarching goal of evolutionary robotics and robotics in general, we would like to deploy autonomous and safe machines out there in the world to work and play alongside us, to do the many, many things that humans would prefer not to do. So we want to create autonomous, capable, and safe machines. What properties would we like those machines to have other than being autonomous, capable, and safe? Cheap. Okay, great. What else? Robust. Excellent. Okay.
Disposable.
Disposable. Great. We've been deploying machines out there in the world for about 300 years, and we're starting to reap the negative benefits of doing that, right? It would be better if we could figure out a different way. If we do want to deploy large numbers of machines out there in the world, maybe we could do it in a better way than we've been doing over the last 300 years. Other features you would like of future autonomous machines?
Scalable.
What do you mean by scalable?
With manufacturing, you're able to make one consistently, or you're able to scale up the size or scale down the size to be used in different situations.
Okay, scale up literally, the size.
Yeah.
Larger, smaller. We might want to scale the rate at which we manufacture things to bring down the cost.
I was just gonna say easy and quick to make, and it kind of goes on the cost.
Okay, that's fine. Manufacturable. What else? How could you make autonomous, safe, capable machines that are cheap, robust, disposable, scalable, manufacturable, and still are not, in retrospect, going to be something you don't necessarily want to put out there?
Fail-safe,
What do you mean by fail-safe?
that the likely modes of failure will not be disastrous.
Okay, okay. Is that something different from robustness? Kind of. So robustness in engineering generally means a machine can keep doing what it's doing in the face of adversity, wear and tear, all the rest. Resilience is a slightly different idea. And the Evil Starfish project that I worked on years back, we called it the Resilient Machines Project because robustness is often not enough. Good news for us, we are robust, but also resilient. You've got a current strategy for how to get through your day, and it works for most days. Tougher days, you kind of tough it out. You keep doing what you're going to do, but every once in a while, you wake up and you face a day for whatever you usually do just is not going to work. So you come up with a plan B. If that doesn't work, a plan C. You get creative about making your way around truly unexpected events. So yes, things that fail well, fail softly, that are resilient would be a good idea. Okay, is that good? Can we go ahead and make machines that have all these properties and we're okay?
I feel like it's still, you can have something that's autonomous and safe and robust, but not reliable.
Okay.
I think there's still that gap where, yes, it can make its own decisions, and yes, it can, do lots of things and in a way where there's kind of fail safes for something that's an activity, but will it do exactly the thing that we tell it to, and is there going to be persistentiation? And it can do all those things safely, but it doesn't necessarily mean it's what we want.
Good point, right? So robustness, resilience, and reliability, they're almost the same thing, but slightly different, right? Hopefully with these 3 R's, maybe we're capturing that sort of thing where we can kind of trust these machines to do the right thing autonomously. That in retrospect, when we see how they adapted to unforeseen circumstances, we kind of look back and say, yeah, that was sort of the best thing the machine could have done. I would have done something similar if I was in that situation. We good? Anything else we're missing?
Would like reproducibility be different from anything? I don't know if it's different. You want to be able to reproduce it like fairly easily.
Okay, you mean the machine itself to reproduce it? Okay, we could add it, let's put it down to manufacturable. We can crank out very large numbers of the same kind of thing. Anything else? Oh.
You want it to be intelligent?
Intelligent, okay, yes, all right. The original 3 desiderata were autonomous, safe, and capable. Capable, intelligent. Again, maybe the same thing, maybe slightly different. You can tell I'm still fishing, right? Okay, we have used state-of-the-art or next-generation evolutionary algorithms, gradient descent. We've designed the heck out of this thing. We now have an autonomous, capable, safe, cheap, robust, disposable, scalable, manufacturable, resilient, reliable, and intelligent machine. Would you eat it? Would you swallow it? Okay. For better, for worse, I see some of you drinking out of plastic cups this morning. You're ingesting some stuff right now that we're starting to learn is maybe not great for us. So, although a little tongue-in-cheek, not quite. What are we missing?
Edible?
Sorry?
Edible?
Edible, absolutely. There is a branch of robotics called edible robotics, absolutely. How about biocompatible in general? Maybe biocompatible fits into, well, not quite disposable. How about biodegradable?
Well, how would you want it biodegradable? Because let's say you want it to work in something that's like a swamp or something. You don't want it to
Maybe you don't want it to degrade. Can we, let's say, can we gain rational control over how biodegradable it is? Depending on where we're going to deploy it, it should degrade under these conditions, not under these. We've spent a lot of time in this class occupying the past. We've looked at what's been done in robotics in the 90s, 2000s, 2010s. We're now starting to think about where we might want to go with robotics in the years to come. And I'm obviously hopelessly biased, but as we're going to see in a moment, my hope is that xenobots are the beginnings of moving in this direction of a new way of thinking about deploying machines. Yes?
I also think that if you're going to be putting it in the body, it needs to be able to navigate within the body to where it needs to go.
Excellent. Okay, so navigatable inside the body. So it's definitely got to be I misspelled that. Biocompatible. You might have something that's biocompatible and biodegradable, but you still don't want to put it inside your body. Why not?
So the other thing, and this might be part of biocompatibility, but the human body tends to consider a lot of things that enter it as threats, and that's what allergies are, and you really don't want to start attacking this and causing a adverse reaction.
Absolutely. So what are we missing? Now we're getting really specific. You actually are going to eat it, or it's going to be injected into you. Now you really want a safe machine.
I mean, all these things also don't, say that the machine, it can do all these things and it can also release radiation.
Could release radiation, okay.
It can biodegrade, I guess, but what products happen when something biodegrades are not necessarily.
Okay. Biodegrade safely. Yes, okay.
Releasing chemicals into the environment.
Talking about like biological material, like made of biological material? No.
Well, we haven't mentioned anything about how we're going to make these robots yet. We're only trying to make a wish list of what we would want for future autonomous, safe, and capable machines. Haven't said anything yet about what we're going to make these robots from, what we want them to do, the fitness function, the objective function, the neural controller. Haven't said anything about that yet. Just what do we want? And maybe what do we not want?
So just to summarize some of the things people said recently is I think we want it to be medically functional.
Okay.
And to do what we intend inside the body and be able to control its behavior once it's been deployed.
Absolutely, right? So we want it to be clinically functional. We want it to do whatever it is we're going to do. You're going to swallow a pill, which isn't really a pill. It's a robot, and it's a Bratenberg vehicle. It's going to be vehicle, what was it? 2B, the lover. It's going to sense something inside the body on its front left and front right, and it's going to swim upstream to follow that signal that it senses inside the body. What might be a signal you can imagine from a clinical point of view you would want it to sense and smell? So there is another branch of robotics called intelligent drug delivery, which is exactly that. Intelligent drug delivery, which is exactly that idea. You swallow something, but it is an active material. It is going to smell or sense the macromolecules, the proteins, the stuff that is given off by a cancerous tumor. When we introduced Bratenberg vehicles at the beginning of the course, I said, imagine a rotten piece of fruit in the back corner of the room. Fruit flies will find it. Lots of organisms are able to sense it. Takes about 2 sensors, 2 motors, and 2 wires in 2 dimensions to create a machine that will move or swim upstream following a source. We could do that here. Maybe this is our clinical target. This is what we want it to do. Find the cancerous cell. The robot goes to that cancerous tumor and then extracts some very small blades and starts spinning those blades to churn up the cancerous tumor. Sounds like a perfectly good solution to me. Maybe not, right? We've seen some hilarious examples of perverse instantiation in this class. It suddenly becomes much less funny when we're thinking about clinical applications of robotics. You definitely do not want an autonomous machine operating inside your body to fulfill whatever the fitness function was in whatever way the evolutionary algorithm found to do so. We haven't solved perverse instantiation yet, so this is still probably sci-fi for the moment, but maybe not for much longer. What else do we need to add to this list to help repress the probability of perverse instantiation?
Off button.
An off button, okay? Yes? Well, what would we call this?
Like a like something that can set like all of the edge weights to 0.
So that's a way of instantiating the big red button, turning it off. Terminatable. The Terminator did something for all of us roboticists, right? You've got to remember somewhere there's got to be a big, obvious red button. When you're watching cool robot videos on YouTube, watch the robots, and usually you can see somewhere on it, there is a literal big red button. If the robot starts doing something wrong, it usually is relatively easy for someone to step in and just turn it off. You swallow something, not so obvious how to make sure that it is easily terminatable. Something definitely we want to have on this list, not so obvious to know how to do it.
Kind of going along with that, you have to be able to communicate with it if you want to be able to turn it off or change its function.
Okay, right. We saw a few lectures about the evolution of signaling and communication. If we can't literally touch the machine or directly touch the machine, how do we communicate a termination signal? Great point. Again, unsolved problem.
Would termination have to be like a button on it or something on it that stops it, or could it be something that like goes after it and kills it?
Whatever it is, it should be either a literal or figurative big red button. What does the big red button mean? Stop, right? Even a non-expert, if they're operating near the machine, they know nothing about robotics, it should be immediately obvious how to stop the thing, right? Pretty much everyone in the Western world that has any bit of education or knows when you see a big red button and you press it, usually the thing will stop, right? But what does that mean for some machines that are a millimeter in diameter and they're operating inside the body? What would be, it's not going to be a literal big red button, not so obvious, not so obvious. Okay, let's come back to something that I think Emily was mentioning. We want to put something inside the human body, and the human body has been evolved for over 3.5 billion years to get rid of intruders. We are considering creating intruders. What else do we need to add to this list? What's that? Oh, adjustable. Adjustable. You mean we would adjust the machine?
Yeah.
Okay, possibly. Would that help us make sure that the body doesn't reject what we're trying to put into it? Maybe. It might help.
Would you have to be able to make the robot using your own organism cells, perhaps?
What's the easiest way to convince your body that what is inside it is you? Make a robot out of you. Okay. So there is an immune challenge as well for the clinical side of this. How are we going to make a machine, let's assume this clinical application that is capable, autonomous, safe, all these things, biocompatible, biodegradable, and your body does not reject it? Now we can start to think about how are we actually going to evolve, design such a machine, and what are we going to make it from? We are probably not going to make it from all the things we've seen in this course so far: metals, plastics, ceramics, batteries, sensors, motors. The thing that the human body, pretty much every other organismal body, hates above all else is very small amounts of metals, plastics, and so on.
This sounds interesting, but how are you gonna get over all the ethical concerns that people might raise, or like all the regulations? Because a lot of people might be completely against
Absolutely. Not only will they be against it, they are already against it. So all of this is science fiction. However, some of it is becoming science fact. So let's go into the how we might do this, and then we'll come back to the ethics. How would we ever get over the social and regulatory hurdles that this vision, this one possible vision for future autonomous machines presents to the public? Okay. All right, there's lots of different ways to introduce the xenobots. Here's one. We've seen this machine already. We've seen a lot of rigid machines in this class already. We want to make the machines resilient. Whatever it is they're going to do when we deploy them out there in the real world, they are going to be faced with surprise. They're going to be faced with unknown unknowns, things for which the programmers or the roboticists did not create contingency plans for. So in the case of traditional robots, if something truly unexpected happens, they have one and only one set of options to adapt to that unknown unknown, which is to alter their neural controller. They can alter their behavior, how they move, given whatever has changed. Soft robots have a different option, which is once they experience something that is unexpected, they have two sets of options available to them. They can adapt their neural controller to try and recover from whatever has happened, which in this case is physical injury, or they can alter their physical structure to adapt. Yeah, they've got two options available to them. So we've looked at rigid machines made of metals and plastics. We looked at soft robots last time that are made from rubbers and silicones, soft, compliable materials. What about a robot that's made from biological components rather than technological components? This is obviously not a robot, or maybe it's obvious. You're looking at a very early tadpole, so a machine, arguably, that's made from a very large number of biological components, which in this case are genetically unmodified cells. Okay. We're going to look at an experiment from two of my biology colleagues, Doug Blackiston and Michael Levin at Tufts. This has nothing to do with xenobots, nothing to do with robotics. We're going to now visit biology for a moment and view biology from the point of view of a roboticist. What would happen if we started to try and build machines to address all these desiderata, but instead of building them from metals and silicone, we're going to think about building them from cells. Why might we want to do that? Because we might get a lot of these things for free. Question?
Are there physics engines where you can simulate biological robots?
There are now, and we're going to see one today. Yes. Okay, so we're thinking about trying to build robots from cells. Cells are an interesting candidate for a building block for autonomous machines. Are they a good choice, bad choice? Let's see. This study, I think, is interesting in that it suggests why you might want to build robots from cells, or that you might be able to build robots from cells. This is a very early frog tadpole. Doug Blackiston has genetically modified the DNA of this tadpole so that it is genetically blinded. It grows from birth without eyes. Ethics has already been mentioned already. A lot of ethical components to what we're going to talk about this morning. Genetically altered tadpole to grow blind. It has no eyes where you would expect eyes to be. But Doug has ectopically rearranged some cells in this tadpole. So ectopic, it means take stuff from one place and of an organism and put it somewhere else. Doug took eye precursor cells. This is a type of stem cell which, generally speaking, shows up in the tadpole here and eventually become functional eyes. But these particular stem cells have been ectopically placed into the tail of the tadpole. Seems like an odd thing to do. This ectopic surgical rearrangement of tissue in this tadpole not only did not kill the tadpole, but as this tadpole gradually developed into an adult frog, the eye precursor cells started to mature and develop into an actual eye, which you can see starting to appear here. This eye started to send out neural processes or synapses that were looking for something. The nerve cord and the nerve tissue in the tail also started to send out synapses, and they connected during development, and as the tail receded, this tadpole grew into a perfectly healthy frog with an eye in its butt. You gotta love science, right? Okay. Not only was this a healthy frog that had no eyes on its front and one on its back, when Doug would wave something at the back, the frog would react in the way you would expect. If it was a fly, it was a picture of a fly, the frog would start to turn around and face in the direction of the fly. If it was something big and shaking like a predator, the frog would tend to move away. A functioning eye in a place you wouldn't expect. How is this possible? Why did this happen?
Plasticity.
Plasticity, can you be a little bit more specific? What do you mean by plasticity?
So the body is actively trying to make something functional, and when it's given an unusual circumstance, it works with what it's got.
Absolutely. So later today, I want you to saw your laptop in half and then ectopically rearrange it. Do the same thing with your car, your drone, your cell phone. Any machine on this planet does not seem to have this capability. So this is just a very dramatic example, but if you think about it, of course, there's examples all over the place that cells are a pretty interesting candidate for building stuff from, because under many circumstances, these cells seem happy, in quotation marks, to be rearranged, which is kind of interesting. So about 5 years ago, Doug and Mike and myself received some joint funding to take this idea and say, well, if you can rearrange frog tissue manually, so Doug came up with the idea to take stuff from here and put it here, could we instead task an AI with coming up with ways to ectopically rearrange frog tissue to come up with something that is different from a normal frog? Okay, that's where the idea for the xenobot started. Okay. All right, so let's talk about the how now. A lot of these pieces are going to look actually quite familiar to you. 5 years ago when we started, Doug Blackiston told us he was able to liberate 2 types of cells from very early frog embryo. So not actually a tadpole, but something that's a little further along from an egg, very, very early frog embryo. He could extract from those eggs a whole bunch of frog skin cells. Like your skin cells, those individual cells or small patches of frog skin are passive and soft. You can pull and push on them and they deform in response to those forces. Doug told us that he could also pull out of that very early frog embryo a second cell type, which are heart muscle cells. These are small cells that when put together, those cells will tend to increase and decrease in volume. Starting to sound familiar? So we decided, let's see if we can get an evolutionary algorithm to figure out how to put these 2 types of cells together to produce something, a machine that is made up of just frog skin cells and just frog heart muscle cells. And we're going to try and get the evolutionary algorithm to figure out how to put these 2 building blocks together to do something. What do you think we wanted that thing to do? What have we seen 1,000 times in this class already? Locomotion. Our old trick, right? Let's try and get something that just walks along the bottom of a petri dish, filled with room temperature fresh water, which is where frogs are happiest. You got a little dish filled with room temperature fresh water, and we're going to put something inside it that's going to settle to the bottom of the dish and then walk along the bottom of the dish. So this is like us trying to sink to the bottom of a swimming pool and then walk along the bottom of the swimming pool. That's what we wanted to do. Yep. So same fitness function we've seen before. We're going to apply this fitness function to an evolutionary algorithm, which in turn is wrapped around a physics engine. Coming back to Abby's question, we're going to see that this is an adapted physics engine for this purpose. Okay, what is the evolutionary algorithm? We borrowed Nick's evolutionary algorithm, which is evolving populations of networks, evolving populations of CPPNs, which stands for compositional pattern producing networks. In this case, everything is more or less the same, but instead of the CPPN placing voxels of 4 different colors inside an empty cube, the CPPN is going to be restricted to only be capable of placing voxels of 2 different colors inside this empty cube. Light blue and these are red-green voxels. We're going to use red-green so that we can see them, these voxels, increasing and decreasing in volume. So far, so good? Questions? No? Okay, so just to recap, our evolutionary algorithm is going to search over the space of all possible ways of placing frog skin cells and frog heart muscle cells to produce something that moves as quickly as possible in about 10 seconds in our frog tissue physics engine. Yes?
Did you find, like, when you were designing this experiment that, like, having soft tissue cells was, like, beneficial to, like increasing movement? Because I know that, like, you were talking about with, like, Nick Cheney's study, how, like, they, like, a lot of the different models, like, evolved out of using soft tissue or, like, hard bone instead of just using muscle.
Great point. So good idea. What's going to happen when we run this evolutionary algorithm? Is it going to discard or is the evolutionary algorithm going to make less and less use of passive soft tissue, which is frog skin cells, and make a big ball of frog heart muscle tissue? Are we going to get the same result as Nick saw with his soft and hard simulated voxels, or are we going to get something different?
Okay.
We're going to see. Who knows, right? Okay, so we don't know yet what we're going to get, but let's pull back again and remember sort of what the big picture of what we're doing. We're taking, although these were genetically modified frog cells to genetically blind the frog in this case, the frog cells are genetically unmodified. These are just 100% natural frog skin cells and frog heart muscle cells. How many of these do we already get for free? No matter what the evolutionary algorithm comes up with, a big ball of muscle, something that's 99% skin and 1% muscle. We already get this, we get this, we get this. There's one thing we are missing from this list. Carbon neutral. These frog cells, as they're growing in the egg, they're absorbing carbon. When these cells eventually die and degrade back into the environment, they break down into things that are mostly carbon. They put the carbon back out into the environment. So if we discount all the compute that we're going to use to design these xenobots and how we actually manufacture them, the xenobots themselves, by definition, are going to be carbon neutral. So far, so good? Okay, so depending on our yardstick, we're already kind of ahead of the game, no matter what the evolutionary algorithm comes up with. Okay. All right, let's have a look at the evolutionary algorithm itself. Nothing too surprising here. We're starting with a population of random CPPNs. I think Sam Kriegman, who was my PhD student at the time, who set up this physics engine and this evolutionary algorithm, used a population size of 100. So at the beginning, we have 100 random CPPNs. you're looking at what the first 3 random CPPNs in that population of 100 produce. We've seen CPPNs a few times in this class now. It's not surprising, this is what they tend to do. So here's one CPPN painting mostly frog skin into this hollow cage and a little bit of heart muscle tissue. This one is producing a big ball of muscle. This third CPPN is painting about half skin, half heart muscle tissue. Okay, there was a question about the physics engine. You can see immediately, or I hope you can see immediately, that this physics engine is 99% VoxCAD. It's what we talked about last time. Voxels where neighboring voxels are connected by a flexible beam, and that beam resists pushing and pulling and twisting and yawing and rolling. Pretty much what we saw last time, with a few additional details put on top. And these are biophysical details, things that Doug knows about frog cells. And these were things that Doug told Sam, who was building the physics engine. Some additional details about how frog skin and how frog heart muscle cells behave. The biophysics of these 2 cell types. You can see that this video looks a lot like the videos we saw last time with the soft robots, but there's a few details that already should look different to you, which are hints about the added biophysical details that Sam added in. What do you think they might be?
Are the cells able to act like extremely independently?
They look like they're acting pretty independently, right? What is the visual cue here that they're acting independently? Independent how?
Like with the different squares activating like very separate to their neighbors. Like it seems almost like there's sort of some pattern, but that may have been like an evolved pattern like over time, but like the individual like cells look like they can sort of act on their own.
Absolutely, they're acting on their own. Which cells are acting on their own? The heart cells are acting on their own. They're expanding and contracting independently. It's a little difficult to see, but the heart muscle cells, when they're green, they're getting bigger, and when they're red, they're getting smaller. So they're getting bigger and smaller in volume, and they're doing so independently. Can we be a little bit more specific? Independent how? What is changing within these cells or within these voxels that's independent from their neighbors?
Seems like the phase of the sinusoidal actuation.
The phase is different, right? Remember when we talked about agonist and antagonistic muscle groups? When one is increasing, one is decreasing. That's a phase offset, right? That one's getting bigger, one's getting smaller, one's getting smaller, one's getting bigger. Things that are in phase get bigger together, smaller together. Here, you see random phase offsets. It's not that there's one group that's always getting bigger when another group is always getting smaller, which is what we saw last time in Nick's soft robotics experiment. Each of the heart muscle cells in each of the 3 robots here has its own random phase offset. Sam deliberately put that into, or added that to VoxCAD to match a particular biophysical detail that Doug communicated to us. So Sam and I were trying to learn the physiology of frogs at the beginning of this project. And so when Doug told us that he could work with heart muscle tissue, we knew enough to know that heart muscle cells, generally speaking, when they're put together, they synchronize their actions. So in your heart, all of the heart cells, and I wish this for you for the rest of your life, that they stay synchronized. All the cells decrease in volume at the same time. They all increase in volume at the same time. And when they're placed together in a shell, in the shape of your heart, all those contracting cells cause your entire heart to contract, which pumps the blood out. And when all the cells expand, your entire heart increases in volume, which pulls blood into the heart. That's what makes a heart a heart. You want these cells to synchronize. Same thing in a frog. As a frog embryo grows into an adult frog, these heart muscle cells go to where they need to go, they connect to their neighbors, and they communicate somehow. Remember our discussions about communication. They need to communicate, and they all decrease together, communicate, and increase together. As we were starting to put this experiment together, it dawned on us that, of course, the CPPN is going to paint or put frog heart muscle cells in patterns and into 3D shapes or distributions that don't exist in the natural frog. So we asked Doug the obvious question, what's going to happen if you rearrange heart muscle cells? Will they still synchronize or not? And Doug said, I don't know. No one's ever done this before. So what should we do? What should we have done at that point when one of the biophysical details is uncertainty? For any one of these ways of arranging frog heart muscle cells, the biologists can't tell us whether or not they're going to synchronize. So what do we tell the physics engine?
Like you want to reward it to synchronize, but I'm guessing you don't just want it to like only be told to synchronize so that it just like synchronizes no matter how that like messes with the other functionality of it?
A good thought. We could try and build synchronization into the fitness function, but the physics engine, the evolutionary algorithm, doesn't know what pattern will produce synchronization except for one pattern, which is an adult frog cell, adult frog heart. So we can put it in the fitness function, but it's meaningless to the evolutionary algorithm.
So what you want to avoid is the evolutionary algorithm exploiting something in your simulator that doesn't match reality.
Absolutely.
If you don't know what's going to happen in reality, you should leave it out or randomize it.
Absolutely. So remember our discussions about the sim to real gap. There's a big sim to real gap here. We're going to try and make robots out of frog heart muscle cells. There is much that is unknown about those cell types, especially when they're rearranged. So the lessons from the sim to real literature that we looked at says, when in doubt, leave it out. Or make it random, or make it as hard on the evolutionary algorithm as possible so that it can't exploit things that aren't going to exist in reality. We do not want this evolutionary algorithm coming up with an idea in which it thinks the cells are going to synchronize, but they won't in reality. So Sam deliberately made things hard on the evolutionary algorithm by randomizing the phase offsets of every frog heart muscle cell that was simulated. We assumed the worst. Assume that these cells, no matter how they're arranged, will never synchronize. So we've given the evolutionary algorithm a pretty hard job. It's got to build a reliable machine, something that reliably moves from left to right along the bottom of the petri dish, but it's going to build that machine out of unreliable parts, partly unreliable. Unreliable in that they're not necessarily going to work together.
In the physics engine, if you're trying to evolve it to work across like the feature which you said water, are you taking into account like the viscosity of the water?
Great question. Are we taking into account the viscosity of the water? Look at these videos. What do you think? There's a pretty strong hint here. It's a little hard to see, but these things are settling slowly onto the featureless white plane. So yes, we reduced gravity, viscosity is a little bit higher. There's some additional details beyond the biophysics, the physics of walking along the bottom of a pool, of a swimming pool. Yeah, so there's a bunch of other subtle physical details that we put on top of VoxCAD, but actually not that many. It's mostly VoxCAD that we're looking at here.
Are the like levels of like these blue boxes sort of indicating like where the water level might be like in the physical world, or like
oh no, that's just the graphics left over from VoxCAD. That's the blue sky at the limit of the featureless white plane. Completely irrelevant, but good observation.
Do you have to feed the cell for it to continuously move?
Oh, great question. Do you have to feed these cells? Okay, hold on to that question. We'll come back to that. Where's the battery coming from? Where's the power coming from to drive all this? We'll come back to that in a moment, in a few minutes. Okay, so here we go. So just again, to think about this design problem, one way to think about this or ground your intuition is imagine you were tasked with building a boat, just a boat that's going to float down the river. You're going to get to place human rowers in the boat. You can have as many rowers in the boat as you want. You can design the 3D geometry of the boat in any way you want. The trick is that when you put these human rowers in the boat, they're just going to do their own thing. They are not going to synchronize, and you want the boat to go straight. A very difficult, non-intuitive design problem, which is exactly the kinds of problems that evolutionary algorithms tend to be good at solving. So far, so good? Okay. All right, here we go. So you just saw the first 3 CPPNs from the initial population of 100 CPPNs. We're evolving these things to walk along the bottom of the Petri dish. You can see that none of these 3 are moving from left to right. If you squint really, really, really hard, you'll notice that this one actually moves about 1 millimeter to the right. This one is actually doing a little bit better than this one and this one. So in the next generation, the evolutionary algorithm is going to delete the CPPN that produced this, delete the CPPN that produced this, and make randomly modified copies of the CPPN that produced this. I'm not even going to show you the video. You know what's going to happen. You're going to start to evolve things that move further and further to the right, like, for example, this thing in the upper left. I don't know if turning off the lights will help a little, maybe a little bit. The little rainbow squiggle underneath shows you how this thing moved. Blue is where it started, and then this red thing moved to the right and ended up here. They're all red. What does that mean? Sorry, this one is the end of one evolutionary algorithm. Sam then rewound the evolutionary tape, created a different population of 100 random CPPNs, ran it for another couple hundred generations, and at the end got this most fit robot. Ran the evolutionary algorithm the 3rd time, 4th time, 100th time. So you're looking at the best robots from 100 evolutionary runs. What happened? They didn't synchronize, so it's a little confusing because they're all shown in red. You see red, what do you not see?
It's all like muscle.
It's all muscle, right? Evolutionary algorithm said, no problem, create a ball of muscle. So we took this to Doug. Doug said, I can't build it. I'm sorry, there's no way. When I'm going to try and actually atopically rearrange these tissues, for various reasons we won't go into this morning, can't do it. I can't make a ball of muscle. So we already failed to cross the sim to real gap. We had manufacturable on the board here somewhere. Doug can't manufacture it. Can't even make it, let alone see whether it matches simulation. So Sam went back and added a term to the fitness function that constrained things so that the evolutionary algorithm had to include frog skin cells. And this is what we got. All of these things in the simulator you just saw, they all move from left to right. We sent Doug all 100 MP4s. We sent him all 100 videos. He went through all 100, said, I can't build that, can't build that, can't build that, can't build that. Turns out that 5 of these 100, Doug said, I think I can actually build these. Okay, we're close. Now, at least we can try to cross the sim to real gap. So far, so good? Okay. So let's watch Doug try and build this one down here. Okay, here we go. Might be a little difficult to see. We've got some muscle on the bottom, and we have some soft skin on the top. Okay. Here's that robot in action. There was a question before about whether the soft passive skin is useful. The answer is yes. How is it useful? It's helping this thing move from left to right.
Is it like a weight?
It's a bit of, yeah, I guess. Actually, maybe yes. Maybe just this passive weight on the top helps, keeping it on the ground. How else does it help? We can see the heart muscle cells on the ventral or the bottom part of this thing, whatever this thing is. And skin on the upper part, the dorsal part. How is the skin helping?
It gives
It gives the heart cells something to hold on to. You'll notice that the heart cells are doing their thing. They're getting bigger and smaller. They're pushing down and back on the floor, which is good, right? That's causing a little bit of forward force, but these are all random forces. These forces are pushing down into the floor, but these forces are also pushing up and into the soft tissue on the top. That soft tissue is absorbing all of those forces coming up from below, and it's kind of averaging them out and supplying those forces back down. So this is a little hand wavy, but somehow the frog soft muscle tissue is averaging the random forces from the motors, from the heart muscle tissue. It's helping. Another example of sort of embodied cognition here. The body is helping with what we want this thing to do. So far, so good? Okay. So Doug saw this video and said, I can build it. Here we go. Okay, here we go. You're looking at a bunch of frog eggs here. Doug is injecting a little bit of a chemical that's going to determine cell type. It's going to make it easier for him to see inside these eggs where the skin cells are and where the heart muscle cells are. This is him pulling off the vitelline membrane, so some of the stuff he doesn't want. So I'm going to skip quickly over a bunch of these videos. This is Doug trying to get at the two Lego bricks, the two types of building blocks that he's going to use to do this. We're looking down through the microscope with Doug. The magnification here is such that these things that you're looking at are about 2 millimeters in diameter. So these things would be just visible to the naked eye. These tiny pliers and calipers are about 1 millimeter across. If you ever meet Doug, do not give him any coffee or Coca-Cola. No caffeine. He's got very, very steady hands. This is not an easy thing to do. Okay. As the title implies here, he's removing the animal caps. There's a lot of biology terminology today. You don't need to remember all this terminology. He's removing about the top 10th of each of the cells, which is where the two types of cell types are that he wants, the skin cells and the heart muscle cells. This is where they tend to be crowded at this particular part in frog development. Tim's still trying to get at the two types of building blocks he needs. Here's two, you're going to see two animal caps in a moment, hopefully. Oh, sorry, 1, 2, 3, 4, 5, 6 animal caps. So he's pulled off the tops of 6 eggs, and that white stuff you see, those are individual frog cells. Gives you a better sense of the scale of magnification that we're looking at here. He's getting rid of the animal caps themselves here and leaving the actual stuff he wants, the frog skin cells and frog heart muscle cells. He's now pulled all of these into a very, very small syringe, and he's now going to inject these cells back into a different petri dish where he's going to actually start building this xenobot here. This dark circle that you just saw, this is a small concave well in the bottom of the petri dish. So all of these cells are settling into this dish. I'm going to skip over this one. This video is sped up quite a bit. You can clearly see the individual frog cells here. Cells, quote unquote, don't like to be alone. There's a little bit of random motion, but they're also seeming to be autonomously clumping together. Imagine trying to build robots or machines out of parts where the individual parts are like helping with the construction. They want to work with you rather than against you. This is preliminary visual evidence already that we're in very different territory here. We're building from components that sort of want to help with what we're doing. Again, scare quotes around all of that. Okay. This is pulled back a little bit. This is about 2,000 cells, again, sped up. You can see the cells don't like to be alone. They're pulling back together. This is later in the process. So cells at this stage in development are quite sticky. Imagine taking a whole bunch of masking tape and rolling it up into balls. So you've got a whole bunch of balls of masking tape and then throwing them into a big concave dish and shaking that dish a little bit. They're all going to glom together into a big ball of sticky spheres. This is sort of the raw material, the block of marble into which Doug is now starting to carve away or remove the unwanted material. You can see this very small forceps on the left, and this is a micro cauterization device on the right, which is a fancy way of saying it's a very, very small car cigarette lighter. It's a very small hot wire that burns away this unwanted material. You should be able to see a little cross pattern here. This is Doug making the 4 little stubby legs in the bottom of this thing. He's eventually going to take this thing and turn it right side up and put it on the ground. I'm showing you a few snapshots from the manufacturing process here. We're skipping over a lot of other parts. Remember that Doug is trying to build this from skin and heart muscle tissue. And there's a particular distribution of heart and skin. We've got heart muscle tissue on the bottom half and skin tissue on the upper half. So when Doug was dropping these cells into this concave dish, he sprinkled down a little thin mat of heart muscle cells, another layer of heart, skin, heart, skin, heart. It's like him making a big hero sandwich. He can layer these 2 different cell types and then scrape away the unwanted material. That, as of 3 years ago, or 4 years ago, was the best way Doug had to try and build what the evolutionary algorithm came up with. So far, so good? Okay. Okay. Here's him putting the finishing touches on things. He's scraping things away, scraping away a little bit of the unwanted material. This xenobot now is about 1 millimeter across. Imagine looking through the microscope at a poppy seed, and with your own hands holding very, very small tools, you are trying to scrape this pattern into a poppy seed. Not an easy thing to do. Okay. Doug, Sam, Mike, and I took a very deep collective breath. Doug took the xenobot, turned it right side up, put it on the bottom of the petri dish, and turned on the camera. And this is what we got. The physical xenobot doesn't quite look like the simulated xenobot. It doesn't quite move like the simulated xenobot, but it was close enough to walk along the bottom of the petri dish from left to right. You probably know by now there are many other xenobots that did not cross the sim to real gap, but as Doug got better at building these things and Sam improved the evolutionary algorithm to evolve these things, we started to produce more and more of them that could cross the sim to real gap. But maybe Doug cheated and put the petri dish on a table that was tilted to the right. Maybe this thing is actually just walking downhill or sliding downhill. How do we know that it's actually the xenobot that's moving from left to right and not some feature of the environment? We were talking about control experiments at the beginning of this morning's lecture. We spent actually one of the hardest parts of this experiment was thinking about what's a fair control. How do we prove that this xenobot is actually walking from left to right because of the heart muscle tissue on the ventral surface and skin on the dorsal surface? We eventually came up with the idea, take the xenobot and place it on its back. If we place it on its back and it still walks from left to right, then it's not the xenobot that's doing the walking. It's something else in its environment that's producing left to right travel. Maybe the table is tilted, or maybe there's a very slight current inside the room temperature fresh water that's pushing the thing to the right. Remember, it's a poppy seed lightly floating on the bottom of a petri dish. Okay, so here we go. You're going to see this is the physical xenobot right side up. And in a moment when the video switches, you're going to see Doug has reached in and flipped the xenobot on its back. Okay, there it is, sorry, on right side up. And in a moment, you're going to see it placed on its back. Exactly the same xenobot in top and bottom. It no longer moves from left to right. So we can conclude from this that the reason this xenobot is moving from left to right is because of its 3D geometry and tissue distribution. Questions, comments? Yes?
How did you know what direction it was facing? Like, I know it's just like, we're just like, oh, excellent, wait, this one, so it's going left.
Yep. Good question. This, actually, from our perspective, left to right, but really move in this direction relative to the xenobot. Remember deictic sensing relative to whom? To the xenobot. That's how. As long as it moved this way, it was fine. If we took this thing and rotated it 180 degrees, you would expect it to move in that direction.
If you were to take this a bit further, because right now you're embedding the controller within like the cells that are also muscle, for example.
Okay.
Like, but bodies don't work like that. You have brain cells. So
Some bodies have brain cells. We need to resist neurochauvinism in this class, right?
Mm-hmm.
Yes, some do.
But if you were to create something that does more complex tasks, then how would you go about that? Would you have to also, if you wanted to create something with brain cells, let's say?
Great question. Yes. Okay. So hold on to that for a moment. I see we have 2 minutes left. So let me spend 1 minute concluding the class and then 1 minute, where is this all going? We have reached the end of evolutionary robotics. I have very much enjoyed my time with you. I hope you have enjoyed the class. Obviously, this isn't the end. We will see each other again on Friday, but I just wanted to take a moment to say I very much enjoyed my time here. I hope you've enjoyed the class as well. Do remember to get everything in on time by Monday night so that Freya can get everything ready for us Tuesday morning. For those of you that are graduating, I wish you all the best in whatever you do next. For those of you that are coming back next year, look forward to seeing you all again. Okay, that gives us 60 seconds to talk about where is this all going. My short answer is, who knows? For me at least, it's can we make autonomous and capable and safe machines that also do all of these? The answer may be yes. The answer may be no. You asked specifically about could we create xenobots in which there is neural tissue inside? I will tell you that we have a very long list of to-do items, things we want to do. Making neurobots is near the top of this list. I'm recording this lecture, so I can't actually talk about things that we've already seen in the lab that we haven't published yet. You can guess what some of these things are. Everything we've seen this morning is making things out of frog cells. You can imagine one of our big burning questions immediately after we saw this was possible is, can you make biobots out of different cell types taken from different organisms? What other organism do you think was at the top of our list about 2 years ago? Exactly. You can go Google anthrobots, anthro for human, anthrobots. Members of the Levin Lab published a paper about anthrobots earlier this year, or late last year. So anthrobots are now a thing, and we would break tons of regulations, but in theory, we can make an anthrobot from you, give it back to you, and you could swallow it and experience no ill effects. Okay, maybe a good place to end today. Thank you all. Good luck with the final preparations on your final project. See you here 7:30 in the morning on Tuesday. You have a quiz due tonight.